The cells of your body that do not give rise to sperm or eggs are called somatic. Oh, come on. The cells of your body that do give rise to sperm and eggs are called germline cells. I'll get rid of this and write it a little bit better. Okay. So, normal cells beget normal cells. Now, sometimes normal cells result in a tumor where these cells are unregulated. Doesn't mean it's cancer. Could be, could not be. If it doesn't invade other cells, it's benign. Benign cells, tumors can be dangerous. They can kill you, but certainly not the sort of danger that normally is imposed on the body by cancer. And we have malignant, which are invasive cells. These can be non-metastatic. That means they invade surrounding tissue, but they don't move. In other words, they don't get into the bloodstream or the lymphatic system, and so are not necessarily as dangerous as metastatic cancers. And then metastatic is traveling cancers. That means they become unanchored and can travel in the lymph or the circulatory system to other distant parts of the body and produce tumors elsewhere. So, we want to look down here at the bottom. Okay, here we go. Okay, at the bottom. And we've got anchorage dependence, cell cycle control, and contact inhibition. And each one of these goes up above, either here, here, or here. So, cancer cells differ from normal cells. They divide when they shouldn't because of loss of which one? If you said cell cycle control, you were correct. Invade surrounding tissues, that's loss of which one? Anchorage dependence or contact inhibition. If you said contact inhibition, you are right. And then moving to other locations, anchorage dependence. We'll call it AD. So, 1 second. Okay, radiation therapy is least effective against tumors that have low lost what? So if you want to know, you should go back to the last lecture on cells division and cancer, and you would see that it's the ability to undergo apoptosis, which is cell so why is it good to take multiple biopsies from a single tumor? Because unregulated cell division leads to increased genetic diversity. That's true, because a given drug may be effective against only one part of the tumor. It's also true. Because a tumor evolution can occur, that's also true. So, I'll say that all of these are true. Oh, it says go to Top Hat. Let's see here. Do I want to... No, I've put up all the answers on Top Hat. You can check that out on your own. So, let's take a look at this. This is mitosis review. So, if we start, we'll start here. Okay, so this is G1. This is where most cells spend most of their time, growth and normal metabolic roles. Now, if a cell is going to go and reproduce itself during mitosis, it's going to go then from the G1 into the S phase, where it replicates all its DNA, so there's twice as much as before. Then it'll go into the second growth phase, so growth and preparation for mitosis, and then finally into the mitotic phase, which includes the prophase, metaphase, which is all the chromosomes. Here we go, come on. All the chromosomes line up along the middle and then pull apart during anaphase. And prophase is when the spindles pull the chromosomes, the duplicated chromosomes, and they line up. And then telophase, which finally ends, and then telophase and cytokinesis kind of happen at the same time, so the nuclear envelope reforms, and the two cells that each have a full complement of the DNA pull apart into two separate cells. So asexual reproduction. Well, mitosis, essentially, you might call that asexual reproduction of cells. A lot of individuals, cells like bacteria and stuff, undergo asexual reproduction where they create clones or copies of themselves. During asexual reproduction, there's no eggs or sperm. The offspring are genetically identical to the parent. In other words, they're just like identical twins. So, back to this again. The cells of your body that do not give rise to sperm or eggs are called what? And what? Well, let's ask. somatic. Okay, so let's talk a little bit about vocabulary. So, somatic cells are diploid. They have each of 2 chromosomes. So, and each pair is called a homologous chromosome. So, these are homologous chromosomes. You get one from each parent. Okay. And then non-homologous chromosomes. And these are different. So if you look up here, these are the 23 or 22 pairs of chromosomes that we have, plus we have a sex chromosome. Females are XX and males are XY. So each of the 22, there's a chromosome that comes from your parents, one from the father and one from the mother. And so right here, each of these comes from one or the other. And they're homologous. They're the same chromosome. They might have different alleles on it, which is different gene variants, but the same genes are on each chromosome. So a karyotype will display chromosomes and what they look like. It can be obtained from a cell at any stage of the cell cycle. As you can see, here on the left is a nice, beautiful karyotype. Here on the right is a less beautiful karyotype. Here you can see the X chromosome and the Y chromosome. God damn it, this thing is terrible. Okay. Diploid and haploid. So, if you have a single one of these chromosomes, right here, that's haploid. If you have both of them, that's diploid. We, meaning humans and other multicellular organisms, are diploid. So, we have our somatic cells have 2 of each chromosome. These are diploid. Each one of those is a haploid chromosome, but they're paired. So we're diploid. We have 23 chromosomes, homologous chromosomes, including the sex cells and the sex chromosomes, for a total of 46. So somatic cells, 2 times 22 is 44. And then we have X chromosomes and Y chromosomes for male and female. X's are XX is female, XY is male. Excuse me. So, here we are again. Our somatic cells have 2 of each chromosome, as I mentioned before, and these are diploid, right? And so they're homologous chromosomes. Human somatic cells, 2 times 22 plus the 2 sex chromosomes. Now, during cell division for meiosis, in other words, for the creation of sperm and eggs, we get down to haploid. And that's why we get one haploid set of chromosomes from our mother and one from our father. So eggs and sperm have one of each chromosome. Example, right here. Okay, this is not a homologous chromosome, this is a single chromosome and it's haploid. There's no homologous pair of chromosomes in haploid, only haploid. Human eggs and sperm, there are 23 total chromosomes. In the diploid state there's 46, 23 pairs. In the haploid there are 23 single chromosomes. So, meiosis. Now, meiosis and mitosis are similar but different. Mitosis just copies a diploid cell and makes a direct copy of itself, a sister copy. Meiosis produced is gametes, and those sperms and eggs, for reproduction, sexual reproduction. So I mentioned you can have asexual reproduction. A lot of all bacteria do it. A lot of eukaryotic cells do it. Some do. Some eukaryotic cells do both, sexual and asexual reproduction. But in sexual reproduction, we have meiosis. So this occurs within the germline and reduces the number of chromosomes by half. Eggs and sperm are therefore, they're haploid instead of diploid. Now, when an egg and a sperm come together, they fertilize. The sperm fertilizes the egg, creates a zygote, this is a fertilized egg, and it restores the diploid number of chromosomes. In other words, 23 pairs or 46 chromosomes total. Excuse me. How would you design meiosis? Well, you would probably break up each of these. Oh, come on. Chromosomes and make copies of them. Okay. Okay. Here we go. Starts like mitosis. Ends like mitosis. So with replicated DNA and separation of sister chromatids. Okay. Okay, so let's take a look. 1 second, I'm going to make some stuff bigger. Okay. So, okay. Now, so mitosis, you duplicate the chromosomes so you have 2 sister chromatids. So here's a parent cell for each. I don't know why this is fucking doing this. Okay. So, This is before chromosome duplication. So here's prophase, metaphase for mitosis. They all line up along the middle. And then finally anaphase and telophase, in which they pull apart and you have two daughter cells. Okay, now in meiosis 1, similar things happen, but you actually have not only chromosome duplication, but a tetrad of two homologous chromosomes are formed at a synapsis. Now, each one of these individual chromosomes for the tetrad is going to end up as a single gamete. The tetrads line up along the metaphase plate. And then anaphase and telophase, you now have these daughter cells, just like before, but they're sister tetrads, right? So they're in two daughter cells. So these are actually haploid cells. Now, there's no further chromosomal duplication in meiosis 2. What happens is the chromatids, which are, remember these things, split apart into single daughter cells, and each one is haploid. Okay? So you have 4 haploid gametes. And that's what happens in meiosis 2. So a gamete will have all 23 chromosomes for humans, but they'll be haploid instead of homologous pairs of chromosomes. They can either be sperm or eggs. So starts like mitosis. There's one special cell division inserted here. Okay. And then in meiosis 2, we end up with haploid cells. Okay. And it's like mitosis 2, and then you have meiosis 2, which gives you the final haploid cells. So, meiosis and fertilization. Meiosis occurs in which kind of cells? Germline cells. Mitosis involves somatic cells, okay? And produces haploid or diploid gametes. Gametes are always haploid. The fertilized egg is called the Okay, so the question is, why have sex? It's a good question. Well, the first reason, of course, that I mentioned in class is that it's fun. But it's actually kind of interesting in evolutionary biology. Asexual reproduction is actually a more efficient way. If you have 10 asexually dividing organisms, and they divide once, they make a copy of themselves, and so they go from, let's say you have 10, you go from 10 to 20. With sexual, you need male and female, sperm and eggs, so if you have 10 individuals, you only get 5 back. So, of course, there's a big debate. Well, what do you get then? Sex is pretty common, especially among eukaryotes. Not all the time. Sometimes they go between asexual and sexual reproduction. But it's still ubiquitous, and the question is, of course, why? Well, there's a bunch of different hypotheses. The ones with perhaps the most evidence to support them is that in environments that are heterogeneous, in other words, different from one place to another, or in time, that gives sexual organisms an advantage. Because through recombination, you can get you can get genetic combinations that in some individuals might perform better in one environment versus another. And in fact, that you can overcome that normally twofold advantage of being asexual. The most common sort of idea for this is originally called the tangled bank, but one example is having hosts being infected by pathogens and parasites. And parasites and pathogens generally can reproduce much faster than multicellular organisms, and so would have an advantage. But through sexual reproduction, we can end up with different variants of combinations of genes that make that individual perhaps more likely to be resistant to a certain pathogen than another. Okay. And this is where we stop. And you can go to lecture capture, and it'll start on from this place right here, and you can follow that all the way through until so back to the lecture. This is the third lecture. The first one I recorded last night. The second one is on lecture capture, and this one which was originally from the 6th, was not recorded with sound, so I'm doing it again. So, if we remember, this is a karyotype for the human chromosomes. There are 22 somatic chromosomes, and then there's a sex chromosome. These are all diploid, so this individual is a female, and then an XY is male. Okay, so each gamete is unique in the sense that there's it either came from the individual's mother or father, but there was also crossing over. In other words, so there was also shuffling of genes from one part of the mother's chromosome to the father's and back and forth. Okay. Now, so sexual reproduction is extremely common in eukaryotes. Some do it, only do sexual reproduction, like our cells. Many others do only asexual reproduction, so that's the eukaryotes. The prokaryotes, in other words, the archaea and the bacteria, only do asexual reproduction. And then there are a number of species that actually do both. And as I went over it before, if you look at it from the perspective of fitness, asexual reproduction is more efficient. If we had 10 individuals and each created one unique copy of itself, there would be 10 copies. So you had 10, now you'd have 20. With sexual reproduction, if you have 10 individuals, 5 and 5, we'll say, in terms of male and female, and you would only produce one diploid individual. So, on the face of it, asexual reproduction wins. It's got a twofold advantage. The thing is, we don't see all that as much asexual reproduction in eukaryotes as we might expect. Primarily because sexual reproduction gives us, or gives organisms, something that asexual reproduction cannot. In an unchanging environment, when you have an asexual reproducing organism, and the environment is stable, and this organism is well adapted to its environment, such a situation will favor asexual reproduction. But in a changing environment, whether it's a heterogeneous environment spatially, in other words, from one location to another, or over time, sexual reproduction permits organisms or populations to adapt to current environments, mainly because of recombination. So, as we kind of learned from Mendel, so sexual reproduction has real big advantages. A good example, this used to be called, well, it still is, the tangled bank. In other words, the environment has so many interactions and is so dynamic that it's difficult for an asexual organism to actually adapt to the different natures of the different kinds of environments that surround it. So, sexual reproduction in that condition is a real advantage. Especially kind of a special case of the tangled bank is our interactions with pathogens and parasites. Now, those are constantly moving targets because pathogens and parasites evolve as well to evade our immune systems. So, having a sexual recombination, you might actually end up with an individual who's got certain allelic associations that not all individuals would have that might make it perhaps better adapted to the current environment, especially when in terms of pathogens. So let's say there might be someone with Ebola who, in an Ebola environment, would actually be resistant to getting Ebola, and that seems to be actually the case. So most people aren't like that, they die, but the individuals that don't leave more offspring. So, in terms of sexual reproduction, there's independent assortment. So remember, we've got individuals who have a chromosome from their mother and their father. Those get sorted independently. And there's crossing over. And so you have 2 to the 23 combinations, about 8 million types of gametes that are available that are unique. random fertilization, in other words, one of those combinations with another combination, means 64 trillion genetically different offspring that your parents could potentially reproduce or produce. Each child is a unique combination of parental traits except for identical twins. So identical twins are essentially clones of each other. Monozygotic twins, there's an embryo that separates into two. They are genetically identical, but there are differences. Once they emerge into the environment after being born, they're subject to different environmental pressures, and there are epigenetic modifications that can happen comparing one to another. Dizygotic twins are just like fraternal twins or your brother and your sister. Full sibs, 2 ovulated eggs, each fertilized by a different sperm. There's no more genetically similar than any non-twin siblings. So, is this sex? We have a roundworm, C. elegans, that is a model organism. It produces sperm, which are stored. It's switched to the production of oocytes, which are eggs, and then the oocytes are fertilized by the stored sperm. So these guys are actually sequential hermaphrodites. They're first male, and then they become female, and then produce offspring. So the thing is, there is gametes here, so we have to remember that. It's definitely sex. So here we have the regular old sexual life cycle. So we have a diploid individual. We also have meiosis. So these are haploid individuals. During meiosis, we end up with 4 individual gametes. Then the haploid cells join and restore the diploid state. Okay, so pretty simple. Here we have a male and a female, and they each produce gametes through meiosis, and we end up a fertilization with a zygote. 2N. So these gametes are 1N. They're haploid. Fertilization produces zygote. Then with mitosis, the zygote develops into a whole organism, in other words, another human, or any other multicellular organism, or single-cell organisms. The only cells that do this are in the germline. So, your somatic cells have 2 of each chromosome. 2. What comes to mind when you think of 2? Uno or die? Die. That means so. These are called what? Diploid. Okay, what are these pairs of chromosomes called? How about number 4? Okay, so as we see here, we've got also the sex chromosomes. So these are somatic cells right here. Oops. Okay, somatic cells, 22. And then we have the sex chromosomes X and Y. In this case, they're haploid. Even though in this individual here, we would call this individual a male because he's X and Y. They form specialized cells during the germline, and this is called, starts with what? An M. And M-E-I-S-O-S-I-S, meiosis. Gametes have each of one chromosome. They only have one. So up above here, we have 2 chromosomes, or homologous chromosomes. If you only have one, they are haploid. Now, this is kind of right, but kind of wrong. So, we'll say that each genome from the parent, the egg, and the sperm, and then the zygote, which is 2N. This is 1N, 1N. Okay, our complete instruction manual to produce offspring, but it's not true. The genome produces proteins, which produce traits and whole organisms, but they only do it in the context of the environment that they're in, whether it's a cellular environment, a molecular environment, an environment that humans walk through every day. All those environments actually affect gene expression. Okay, so the egg has 23 pages of, or 23 chromosomes from each egg, and 23 chromosomes from each sperm. And when they fertilize, each of those 23 pair up with its like-minded brother or sister, and that produces a fertilized egg and a 2N individual who has 46 chromosomes, 2 pairs of 23 each. Excuse me. So, the hemoglobin gene, we've been looking at this. This is what it looks like. All these base pairs correspond to one of the 4 bases. Each 3 base pairs corresponds to a nucleotide made of or an amino acid. So these 3 pairs are called a codon, and they equal amino acid. So this is a normal hemoglobin gene on chromosome 11. It's got this many base pairs. You'd have to count them. I don't know how many, probably 1,000 or something, more than that. And then we also have, so here we are, gene, but we have S, which is the abnormal type. Now remember, the S, when you have an individual who is HbA, HbS, this individual is a carrier for the S allele, but they're also resistant to malaria. However, when you get HbS, you get sickle cell anemia, and that's fatal. And, well, it's usually fatal by, let's say, by teens, but they're working on it. So it's a pretty high cost to pay for being a carrier but being resistant to malaria. In other words, being resistant to malaria would actually have higher fitness than losing all those homozygote recessive individuals. However, in a malaria-free zone, there's a price to be paid. First of all HbS individuals have 0 relative fitness and absolute fitness, we'll say. And it turns out that HbS individuals, even though they express what appears to be the normal genotype of hemoglobin, the actual hemoglobin gene does have slight sickling and so is less efficient at carrying oxygen. And so they have some of the same problems that full-blown HbS individuals have, but not to the same extent. So there's actually selection against both the homozygous recessive, very strong selection here, and a little bit here. Here's the normal. Okay, so here we have the normal hemoglobin gene. Each one is normal, right? And then we have the clumped hemoglobin gene. And this is the one that causes problems. Minus, minus, minus. Okay. And you can see where the problem is. In this nucleotide amino acid sequence, we have leucine, I don't know, provaline. Anyway, the difference here is on the 4th one. You go from glucine, which is the normal part, to valine, which is the abnormal one. And this is what the phenotype looks like. normal is this one. The sickle cell type is this one. And if we were to make kind of the HbA/HbS individual, it would be very much like this one, except it might be a little bit less circular and wouldn't carry oxygen quite as well. So, genetics vocabulary. Heterozygous. In this case, we have two different alleles, HbA, HbS. This individual is resistant to malaria, and is a carrier of the HbS allele and can pass it on to their offspring. There's a 50% chance of passing it on to their offspring. Now, there's also homozygous. That's where you have two of the same allele versus different. So, okay, these are homozygous. This is normal. We call it wild type. And these are the recessive genotype and phenotype. So normal right here is genotype and phenotype, which is normal red blood cells. And this is abnormal. Now, however, HbA, this one right here, is dominant. So when the heterozygous form shows up, it has basically the normal phenotype. Okay, so here we are. We're going to look at it. This is the genotype. Over here is the phenotype. So, what would you say? Would HbA be normal or abnormal? Well, let's start here. Would HbS be normal phenotype or abnormal phenotype? Abnormal. Would HbA/HbS be normal or abnormal? Almost normal. And then HbA would be normal. Now, so here we've introduced a kind of new concepts, not really, but just to go over it. A genotype. That's the alleles that you have. So the base pairs all strung together that code for amino acids. And then the phenotype, the actual physical trait. Now, physical in the sense that it can be a trait that's expressed during adulthood, during development. It can be behavioral trait, any sort of trait like that. Genotypes do not code for phenotypes. They code for proteins, which then gives you the phenotype depending on expression patterns. In this case, heterozygotes they don't show the disease, sickle cell anemia, but they carry the allele. However, they're resistant, remember, to malaria. In a malarial environment, they have higher fitness. In non-malaria environment, they have slightly lower fitness than homozygous. Okay. So let's look at this. Okay. If allele A1 is dominant to allele A2, in other words, we'll say HBA versus HBS, then the dominant phenotype is expressed in both the homozygous and heterozygous genotype. Okay. A phenotype that is seen only in the homozygous, in this case A2, which is only expressed as a recessive A2A2. I want you to understand, though, that this is homozygous recessive. Not all, most recessive phenotypes are not disease phenotypes. Just because HBA and HBS that we looked at before and HBS are associated with sickle cell, most recessive alleles are not associated with disease. They just only express themselves when they're together. The dominant allele has nothing to do with the frequency of an allele in the population. In other words, because something is dominant doesn't make it more common or more rare. As we'll see, there are certain genetic diseases that are very rare that are dominant. In other words, you have one copy and you get the disease, and they're very rare. Now, in terms of the phenotype, there will usually, if there's a dominant allele, then the dominant phenotype will be more common unless, let's say, there's selection against it. In other words, if there's selection against a dominant and favors a recessive, then it wouldn't be that way. So here's some examples in terms of dominance. Freckles, tongue rolling. I can't do that. I don't have freckles. I don't think I have dimples. No, I don't have a widow's peak. Looks like Paul Ryan. What do I have? I think I have detached earlobes. I'm dominant. Amazing. Okay, now, recessive traits. In other words, only happens when there's two of the same allele in the genotype. Let's say A1, A1. Okay, cleft chin, hitchhiker's thumb, attached earlobes, I'm better than him, and then albinism, which is a recessive genetic disease. In other words, you don't produce pigment. And there's also other things associated with various forms of albinism that are not so hot. There was a great blues guitarist. He died away a little bit back. His name was Johnny Winter. and incredible blues guitarist, and he was albino. But he managed to live until he was, I think, 70 or something. So, meiosis produces gametes with either HBA or HBS, if we're back here, to sickle cell. If it's not sickle cell, if it's just recessive, then they produce gametes that are either normal or recessive. Or not normal, wild type, we'll say, and recessive. Okay. So half your gametes will have HBA and half will have HBS. Right? Here's some sperm. Remember, they're a haploid, not diploid, so they carry one copy of each chromosome, all 23 of them. Or, and this is the HBS variant. The idea behind this is the same as for your STR analysis. Remember, single repeats as for the possible outcomes. So I'm going to go through it. We'll see what it looks like in the end. But we have mom. And we also have mom here, and then we have dad and dad, or we have mom. Hello? Okay, back to where we are. So, we have mom there, and we have mom here, and then we have dad here and dad here. So, these are the four possible outcomes. So, here we are. Okay. So, predicting outcomes. Now, we'll go on to the next one where we'll see what a Punnett square looks like. In fact, we can do it right now. Okay. What we do is we put each allele of each individual along either the left side or the top. So, if we have two heterozygotes, this individual will be HBA, HBS, and along the top we'll say HBA and HBS. So, HBA, HBA, we have one. And that's 1/4. So the whole is 1. This is equal to 1/4. Go along with this individual right here, and we go to HBS. Then we have an HBA, heterozygote HBS. That's also 1/4. Here we have HBS, HBA, 1/4. Now, this is the same. as this. These are heterozygotes, so we basically have HBA, HBS for the heterozygotes. And if you add 1/4 plus 1/4 equals 1/2 heterozygotes. And then for this last one, we start with HBS and HBS, and we end up with HBS, HBS, which will not breed, and that's 1/4 as well. So, this is what it looks like again. HBA, HBA equals 25%. HBA, HBS equals 25%. And 25%. HBS, HBA, which is the same. That's 50. And HBA, HBS, HBS is 25%. This is the simplest case, where there's one locus, that means one gene, and two alleles. There are oftentimes many loci associated with a trait and many different alleles. So, some genetic diseases are attributable to defects in a single gene. So, sickle cell anemia, cystic fibrosis. Some are caused by a dominant gene. That means all you need is the Huntington's allele H instead of wild type, and you'll have the disease. Most diseases, and in fact most traits, are influenced by many genes or several genes. Okay? So, forget about diseases, just traits. Most traits are polygenic. There are many cases of diseases that are found that are polygenic as well, like high cholesterol, schizophrenia, cancer, number of different things. Okay. So, cystic fibrosis normally transports chloride ions across the cells of the lungs. If this protein is not functioning, a thick mucus layer builds up in the lungs and also in the stomach and intestines. How many CFTR alleles are needed for a person to be a carrier? Remember, this is a recessive. So recessive, you need 2. How many, sorry, my mistake. To be a carrier, all you need is 1. Okay. In other words, C plus is the wild type, C minus, that's the heterozygote, is the bad part, and these are carriers. To actually have the disease, you need C minus, C minus. Okay. Just like before, carriers are heterozygotes. So, in this case, we've got, okay. We'll say CN and CM for mutant. Okay? So here we have CN, CN 1/4. Here we have CM 1/4. These have cystic fibrosis. And then CN CM. And then CM CN, which is the same. So 50% are carriers, these guys. This individual is normal in terms of phenotype, and this one is abnormal. If a man with CF marries an unaffected woman whose mother had cystic fibrosis, what is the probability that the child will have the disease? So here's the man. He has it. You need 2 alleles of the mutant. To have it. So he's in bad shape. He marries a woman whose mother had cystic fibrosis. So if her mother had cystic fibrosis, she's actually got a 1 in 2 chance of carrying the allele. So we'll say here the woman is N for normal and mutant for here. Okay, so in this case, the probabilities are CN CM, carrier, not cystic fibrosis, CN CM, same, normal, and this is no, this is not CN, it's CM, sorry. Remember the mother, 50% chance, so normal, abnormal. And here we have CN, CM, no, CM, CM, and CM, CM. So 50% cystic fibrosis. Huntington's disease, it's a very rare dominant allele, and it's a neurological disease. In other words, it's a neurological degenerative disease. You get an abnormal protein. Usually onset is later in life, let's say 40 to 45. However, there's something called genetic anticipation. You don't have to know this, but this disease is caused by extra repeats in a region. Instead of having a certain number of repeats, let's say less than 12, this individual has more than 12 and will show Huntington's disease. Now, the trouble with anticipation is it's a unstable repeat, and so each generation it generates more repeats and therefore worse disease, earlier onset. So here we look. Your father has been diagnosed with Huntington's disease, a dominant condition. What are his chances of getting it? Well, let's do a Punnett square. Okay, here's your father, let's say over here, and he's got H, normal, NHM. Okay. Now, so, and then of course your mother will say is HM, I mean normal, H normal. So, here we have HN, HN, say N, N, normal. And here we'll have HM, HN, that's disease, Huntington's, and the same thing here. So, what we have is 50% normal, and they're not carriers, and then 50% individuals with Huntington's disease. So we'll go through this again. Just pay attention. HH. Okay, so we'll do woman, man. Man is normal or abnormal. And normal. Woman is normal and normal. And okay, so abnormal, oops, abnormal, abnormal, and then, so this is Huntington's disease, and this is normal. So, Mendel was the father of genetics. He discovered how genetics is actually particulate inheritance. In other words, we have alleles, let's say HBA, HBS, or A1, A2, A3, A4, and not like at the time, which was thought to be blending inheritance, which was like be mixing red and white paint, you get pink, right? So with blending inheritance, it doesn't really work with evolution, and it was a big problem for Darwin because they didn't really know anything about particulate inheritance. It turns out Mendel was off by himself. He published in an agricultural journal. Darwin never read it. But in Mendel's own journals that he got, he actually had he had a copy of Darwin's On the Origin of Species, but he didn't quite recognize how it pertained to evolution. Evolution, because it's particulate inheritance, evolution is actually allowed to work. Otherwise, you get things washed out, like red and white, with particulate inheritance. Like, let's say, you might have red as dominant and white as recessive. You would get red for the homozygous, red for the heterozygote, and then white for the homozygote white. So, family pedigrees. Okay. Now, we don't know. We have this dominant trait. Recessive trait. Okay, so here we are, recessive, and we have questions about what this is, about what this is, and about what this is. Okay, now here, these little dots right here mean they're carriers. Okay, so what we have here is we've got this carrier was derived from this individual. Of course, it could be this individual, but it's probably not. And so we have 1 and 2 that are normal phenotype. This is the recessive phenotype. Same thing here, we don't know. But if we have 1 and 4, then it's probably that they're both heterozygous. In other words, they're half and half. Okay. And so if we follow this down, the only way we actually show the trait is if in 3 and 4 we get the carriers mating and you get a homozygous recessive phenotype. So these are all somatic cells. They're autosomal or somatic. They appear equally in both sexes. They have nothing to do with sex chromosome. Mistakes in meiosis happen. So, one is failure of chromosomes to separate during cell division. Okay. Let's go. Non-disjunction, failure of chromosomes to separate during division. It results in gametes with one too many or few chromosomes. Most of these will die at birth. In other words, they're dead. But sometimes, here we have trisomy, which is Down syndrome, where an individual has 3 of one chromosome, none of another. Okay? So, Down syndrome depends on expression patterns. They look different, but some of them are either not retarded or mildly retarded or very retarded. In terms of sex chromosomes, so we have 23 pairs of chromosomes. 22 are normal autosomes. Both males and females have the same. Sex chromosomes, 1 female and 1 male equals a male. Okay? Or if the male donates an X, then it's female. So if the male donates this individual to this female, either one, they end up with female. If the male donates this to either one, then we have a male offspring. So, as I mentioned, an X and an X, female. XY, male. More mistakes in meiosis. So, presence of a Y confers the ability to develop male characteristics. So, we've got a female genotype, XX is normal. We have a male genotype, XY, that's normal. XO, in other words, kind of only an X, is Turner syndrome. Klinefelter syndrome is XYY. And XXX is a superwoman, XXX. So, here's our sex chromosomes. An X equals F, Y equals M. Now, when you put two X's together, F. When you put an X and a Y together, male. Okay. Now, the heredity of sex chromosomes is slightly different. With autosomal somatic cell mutations, if there's a mutation in one chromosome, like let's say HBA, let me put in a new, okay, let's look at HBA again. Okay, so these are autosomes or somatic cells, both individuals, has nothing to do with sex. Okay, so let's say there's an abnormality in the B, and we end up with HBA, HBS. And if A is dominant, you put these together, you have a normal, almost normal phenotype, right? So only when it's HBS, that we have abnormal. Now what's happening here is something called rescue. So if you have rescue, in terms of recessive, the normal type will produce enough protein to mask the abnormal type. Okay. In sex chromosomes, it's not so simple. Well, this is simple. What determines the baby's sex? Mom or dad? Question? Dad. He either donates an X with the mom, or he donates a Y. XX, female, XY, male. Okay. These are sex-linked genes. X-linked located on the X chromosome, Y-linked located on the Y chromosome. Males get their Y from who? Dad. And their X from mom. Can males be carriers of X-linked traits? We'll see why not. Males have X-linked traits, but they're not carriers. They're carriers in the sense they do, but remember, they don't have rescue from a Y. So, why do some genetic conditions affect males more often than females? A male has a single X chromosome, so he has only one copy of these genes, not two. So in other words, if you had two, there might be rescue, like on an autosome, and you wouldn't see the phenotype of the recessive condition. A female has two copies, so she can carry an X normal and an X mutant and show normal phenotype, but pass on XM to the male who has a Y. Y cannot rescue M, so the individual XM actually has the trait, the mutant trait. So, in this case, red-green color blindness is caused by a recessive X that is only donated, well, we'll see. If a colorblind man marries a woman who is a carrier for the recessive allele, what is the predictive outcome for their children? So, colorblind man is X, Y, and the X is a mutant. Okay, so, and a woman who's the carrier and also has an X normal mutant. If we do a Punnett square, let's go to the next page. Okay. Okay, so we'll say the man is colorblind, and he's got an X, and he's got a Y, and the kind of X he has is recessive, so he's got the mutant. And the woman, this is the man up here, woman, and she's got an X and an X. One's normal, one's mutant. Okay, so, because she's heterozygous and a carrier. So, when the normal meets a mutant, you've got a carrier, XN, XM, right? And you've got an X normal and a Y, and so this is a normal male. Here, you've got a double XM, which is probably lethal, we'll say. But let's say here we have an X mutant and a Y. And so this is colorblind. Hemophilia is caused by a recessive X-linked trait as well. Female carrier marries an unaffected male. What fraction of their sons will be affected? Do it again. Okay. X, N, X, M. And then we have, for the male, X, N, X, N. So, it'll be 50-50. 2 XNs, XNs, that's 50%. And here, XM. And these are hemophilia. It's called the royal disease because the royals were excessively inbred in Europe, and an individual had this disease, There was a mutation that started it all, and because they inbred, they passed it along, and they had a higher than normal incidence of hemophilia, along with a bunch of other recessive deleterious alleles from inbreeding depression. So, if in terms of color blindness, now, one thing I want to mention, why is it that it's all the males that get the bad disease and the females don't? They just carry it. Well, it's simple. Males are the heterogametic sex. So, heterogametic sex. So that means that as a heterogametic sex, for the sex-linked chromosome, if you inherit a bad X, let's say for a disease, and you also have a Y, that individual is going to have the trait, the deleterious trait, and a lot of times it can affect, let's say, color blindness, hemophilia, sterility, all sorts of different things. Whereas the homogametic sex, that's the female, XX, they can only be carriers and donate one. But if they have normal, everything's good and it doesn't really matter because there's rescue effect in terms of the protein made. So, males, let's see. Well, let's see. Males. Well, if a male can still breed, they can be a carrier. But anyway, if you're a colorblind male, in other words, you have an XM and a Y, which parent gave you your color blindness? Well, the Y came from who? Dad. You can only get the Y from Dad. So the X must have come from Mom. Now, in mammals, for the most part, the male is a heterogametic sex. In other words, there's a sex chromosomes, XY. The males have the Y. The females have two X's. Now, it turns out it's not that way in all animals or all organisms that have gender. In birds, in fact, it's different. It's ZW, and the heterogametic sex are females, and they are ZW equals female, and the heterogametic sex, or the homogametic sex, are the males, ZZ. So, in birds, it's the females who suffer, and they get donated from their dads. In other words, it's all because of their old dad. Now, these are recessive traits. Okay. So, qualitative traits show this or that phenotypes. In other words, if we have a gene that gives pigment, we'll say, and the pigment, it's probably anthocyanin, it's a pigment-producing gene, is purple, okay, or white flowers. It has this or that phenotype. This is often, usually because of dominance. So we have a P and a white, and you get P for phenotype. You have a P and a P, get P, or white flowers You have a WW, and you have W. Okay. Cystic fibrosis, or not. Attached earlobes, or not. Okay. These are called qualitative traits. Okay. So, qualitative traits again. Here we are. Okay, this or that. Now, quantitative traits are different. They show continuous variation, kind of like blending inheritance. So let's say flower color was actually caused by many genes, each of which has small effect. You would have a continuous distribution of color, right? From white to blue, let's say, and light blue in the middle, something like that. Okay, but so this is not qualitative, this is continuous. You have a normal distribution. And height is usually many genes that affect height, many genes that affect skin color, musical ability, cholesterol levels. Here we are again. And here's a normal distribution. So this is either purple or white. And this is, if it was actually not height, but let's say it was color, you'd have, let's say, light color, and then dark color, and then gray color all over. And so height is like that, skin color, musical ability, all that. Traits, so traits that are controlled by a single gene are called monogenic. And under many genes, well, instead of mono, we have polygenic. Which is more likely to show continuous variation or continuous variation. Polygenic. Excuse me. Okay, so eye color, pigment types. There's 3 different genes. And so you have different distribution of pigment. Now, that's why you sometimes see all different colors of blue eyes. Some are light blue, and some are dark blue, really dark blue, we'll say, versus light blue, a little more blue-green. Okay? So, eye color is a polygenic trait. And we end up with a wide range of eye colors. It's not like blue or brown. It usually different colors of blue, different colors of brown, dark brown, light brown, green, things like that. Now, as I mentioned earlier, this idea that there is that traits are caused by genes or traits are caused by environment are, in fact, That's an incorrect model. Traits are, in a sense, they're necessary to have a genetic component, but not sufficient to produce a certain trait. You also need a certain environment as well, in terms of gene expression. So, traits are controlled by genes and highly influenced by environment. In other words, necessary, give me this, but not sufficient. For it to be sufficient, you need the environment. Necessary, but not sufficient either, for actually proper gene expression. Heritability. How much of a given trait is due to genetics? It's a measure of a relative importance of genes in determining a trait under a certain set of conditions. This is important. Under a certain environment. Okay? In plants, in these trees, there's a Krumholtz variety of many different species that live at high elevation. They're kind of gnarled and bent over, compared to what we would consider more normal growth, straight up and down. Now, the Krumholtz variety is simply a matter of gene expression that those genes would not be expressed without the effects of cold and wind, and then that leads to a subsequent phenotype. So, we have a tree. Okay. And under normal conditions, okay, we have a tree. It's got all that stuff, right? And then for Krumholtz, we've got a tree, and it's all bent over and very thick at the base, so it won't be blown over. This is only found at high elevations where it's windy and cold. At lower elevations, we get a normal-looking tree. There's variation in this normal tree in terms of height, and let's say, but basically normal up and down. So, in heritability, we look at the importance of genes, determining a trait under a certain set of conditions. We can oftentimes discover more about heritability by looking at twin studies, identical twins versus fraternal twins, or brothers and sisters, siblings. Okay, so these are identical twins. Do they act exactly the same? No, but it's highly likely that they'll have similar levels of intelligence if they're brought up in the same environment, and dancing ability. Okay, now, if they were fraternal, not necessarily. Just because of genes, one might be more intelligent than another, less intelligent, or whatever. So we can oftentimes see how important genes are in terms of intelligence. So let's say intelligence is highly heritable. We would see that intelligence in similar environments between these two twins would be nearly identical. But in fraternal twins, we would probably see, just like in siblings, there'd be differences. So, how do genes matter? For quantitative traits, it is difficult to predict the phenotypes of children from the phenotypes of parents, mainly because polygenic. we don't know. There's a lot of genes involved, and it's hard to say which alleles are being donated from each parent. Genes can have a very strong influence on even the most complex traits, behavioral or otherwise. But environment can have large effects even on traits under genetic control, kind of like I explained earlier a long time ago in terms of how hearts develop. You have certain developmental genes, but the heart develops from the circulatory system that goes first, and then because of shear forces turning genes on and off between anterior, dorsal, all that kind of stuff, you end up forming a heart under the right cellular environment. And I believe that's it.